right, hi you guys. So welcome to our first 1802 lecture online. I'm Georgia, I'm one of the TAs, and I'm going to be lecturing to you guys about fungal diversity and life cycles. So just a quick note before I start lecturing, I'm going to be doing this in 5 different activities, and each of those activities correspond with activities in your handout and also questions in your worksheet. So with each activity, there'll be a section of the PowerPoint, and you can kind of scroll through and see how the activities are broken up in the PowerPoint file. And there will also be some extra supplemental videos that I'll post that are mostly YouTube videos that some are, I think, pretty necessary to answer the questions, and some are just kind of extra. So I always encourage looking at things and hearing them in many different ways. And I know that's going to be really challenging with it being online. So if you have any questions, please feel free to reach out to your lab instructor or me if you feel like it. Really anybody. Will just make sure that we can go over with this with you and you guys feel great about it because fungi are super cool. All right, so we're gonna just start out with our exercise overview. So there are still learning objectives. So we're gonna talk about really general properties of fungi and understand the zygotic life cycle. And then we're gonna go into different taxonomic groups, Zygomycota, Ascomycota, Basidiomycota, and those are all different fungal groups. And we're gonna talk about their life cycle, their traits, things like that. And then we're gonna talk a little bit about lichens at the end. Okay, so this is the start of activity 1. So this is just going to be a quick overview of fungi and Zygomycota. So for this activity, make sure you listen to this whole lecture, maybe take notes, and then there'll be some supplemental videos posted on the As You Learn site that say activity 1 as well. And then these are going to be the materials you need to answer questions 1 and 2. All right, so the kingdom fungi is a very highly diverse, really unique kingdom of organisms. They exhibit eukaryotic cells, and the cell walls, they have cell walls and they contain glucans and chitin. So some people say chitin, I don't know, people say it differently. I say chitin, but it's the same thing that makes invertebrate shells really tough. They're really long chain polysaccharides, so they're really hardy. They come in single and multicellular forms. We'll talk about some examples of those. And they are heterotrophic, which means that they require a carbon energy source. So they cannot, like plants, make their own food. They have to get food from other things. They can produce both sexually and asexually through spores, and their life cycle is a zygotic life cycle. Okay, so here is the ancestral tree. We can see where fungi fit in on here. So fungi and animals are more closely related. They have a common ancestor of chanoflagellates that we talked about in the microorganism week. So fungi are more closely related to animals. You can see that they are pretty far away over from the land plants that we talked about in the past couple weeks. Okay, so if we break up the fungi group a little bit closer, we can see that they are broken up into many groups. There are chytrids and zygomycetes, and those have zygosporangium with zygospores. Then we're going to talk a little bit about glomeromycota just briefly, and then the 2 other major groups are Basidiomycotas and Ascomycota. Okay, so these phylogenetics, chytromycetes, or chytrids for short, these are the most ancestral fungal form. You'll probably have heard about these in the news, maybe about amphibians. We'll talk about them a little bit more closely. Zygomycetes are bread molds. Glomeromycota are mostly arbuscular mycorrhizal fungi. They're the fungi that have the association with plant roots. They all fall in that group. And then we have 2 really large groups of Ascomycota, which are sac fungi. This is the largest, most diverse group of fungi. And then Basidiomycota, which are club fungi. These include a lot of other really well-known mushrooms too. Okay, so the zygotic life cycle that all fungi exhibit, a majority of this life cycle is spent as 1N. But we don't only have 1N and 2N in this life cycle, we also have a middle ground of N plus N that I'll talk about in the next slide. But just like we've seen before, you have meiosis that creates the spores, and the spores will be of different mating types. And so we'll talk about that in the next slide a little bit too, but they'll grow via mitosis to a 1N individual that'll be multicellular. It'll be all one mating type. And then that 1N individual will produce gametes. The gametes will come together and go through fertilization to create a 2N zygote. The zygote usually goes under meiosis pretty quickly. Fungi don't really stay in 2N for very long at all. actually. And we'll talk about the fertilization step in fungi is where it gets a little bit challenging and different than what we've seen in plants or maybe what you guys know with animals. Okay, so there are two main parts of fungi that we're going to talk about: the vegetative body, and so that's like the main body of a fungi. So most fungi are multicellular organisms, and they have hair-like structures called hyphae. This is the main growth of the fungi. When there are many hyphae and it grows into large mats, so much so that you can see them with the naked eye, they're called mycelium. Hyphae usually grow within a substrate. It's usually soil, but it can be all sorts of different things. So bread mold hyphae grow on bread. There's a lot of other fungi that grow in dead wood or other rotting material. And so hyphae is what most of their body is made up of, even though it's usually kind of hidden and sometimes really thin and hard to see if it's just a single strand. There are two main types of hyphae. There are aseptate and septate, and those are these kind of separate some large groups of fungi. So aseptate means that there are no cell walls to divide the nuclei, and septate means that there are cell walls to divide the nuclei. And we have a YouTube video that shows mycelium growing in a medium. It's a time lapse. You guys should check it out. So just if you're thinking about the time spent of a mushroom, most of their life is spent in the vegetative body state, just hanging out, doing their thing. Okay, and then the sexually reproductive part are the mushrooms that what you think of when you think of a mushroom. So that's the fruiting body. These are usually what grows above ground, and they all have a layer on them called the hymenium, and the hymenium is the reproductively active layer of the fruiting body. So really, I think a lot of people, when they think of mushrooms, they only think of the fruiting body, but that'd be like only thinking about adult humans and thinking of only their sex organs, and that's how you would identify them. Really, there's a lot more going on in their whole body, but there is one layer, so that hymenium will look different in the different groups that we'll talk about. So that word will come up later. Yep. All right, and then like I said a little bit ago, fertilization is a little bit weird with fungi. So fertilization happens in two stages. So there's plasmogamy and karyogamy. So the plasmogamy is when the cytoplasm of two mating type parent cells fuse together. And at this point, the nuclei are not fusing together themselves. They're remaining separate. And so this makes two haploid nuclei close together in the same cell. So we call this N plus N. So it's two 1N organisms that are coming together in the same cell, but they're not fusing their nuclei. So they're not quite 2N yet. So we had these 1N organisms here, and they're of different mating types. So instead of having like a male and female part of fungi, it's mating types, and some species of fungi can have hundreds of different mating types, and so it makes all these different interesting combinations. But for the class, we'll just talk about a simple way of having a plus and a minus. So plus mating type comes together with minus mating type, and they make an N plus N cell. So you can see how there's a dikaryotic cell right here, and that's some of the blue nuclei and some of the red nuclei before they have fused together. So there's still there's still N plus N here. And then karyogamy is when those two nuclei are going to fuse together to form a specialized diploid structure. Okay, so the diploid structure is 2N that you think of normally. But so when we talk about fertilization, in the life cycle, just know that for fungi, fertilization is made up of plasmogamy and karyogamy. So you'll have to put that in your life cycle diagrams. It's not as simple as writing that the gametes come together and it's fertilization. Sometimes a lot of fungi like to hang out in N plus N for a while. So let's just talk about that one more time. So you have two different mating types. So this could be a plus and a minus that are both 1N mature structures that come together via plasmogamy, and they hang out in N plus N for a short or long period of time. And then via karyogamy, the N plus N fuse together to make diploid nuclei. It's 2N, and that's within a sporangium now, or spores. And then it'll separate it's not spores yet here. So it's in the zygosporangium, but then those diploid nuclei are going to go through meiosis to become haploid nuclei that are then going to be the spores. So show that here. Fertilization, which is plasmogamy and karyogamy, are coming together, making that 2N zygote. 2N zygote is going to undergo meiosis really quickly, happening in the zygosporangium, and then it's going to break up into those 1N spores of different mating types. Okay, so sexual reproduction in fungi, just like we saw in plants, there are some pretty specialized names for each group. So kind of get in the habit of using these different names rather than saying just sporangium and spores. Talk about the most specific that you can. So meiosis is always going to occur within specialized sporangium and produce specialized spore, but it's going to be a different name in each group. So in zygomycota, which are bread molds, you're going to have a zygosporangium that contains zygospores. In ascomycota, you're having an ascus or asci that contains ascospores. And in basidiomycota, you have a basidium that contains basidiospores. So I know it's kind of weird terminology, and fungi sometimes seems like you're speaking a new language, and you are because it's science. So I'm just kind of getting the habit of using those new words. And asexual reproduction in fungi also happens, but it's just involving mitosis to only form spores that are just exact copies of the parent organism. All right, so that is all for activity 1. So go ahead and take a pause, and you're going to answer questions 1 and 2, and then you can watch those other YouTube videos that I've posted as well, and then you can tune in to the next lecture.